This video and its accompanying supplemental guide summarize methods developed at the EPA Office of Research and Development, Narragansett Laboratory, for estimating the chronic toxicity of marine or estuarine effluents and receiving waters on the sexual reproduction of the marine macroalga Champia parvula. Males and females are exposed in a static system to effluents or receiving waters for 2 days, followed by a 5 to 7 day recovery period for the female plants in a control medium. Cystocarp production by the female, which indicates sexual reproduction, is used as the test endpoint. The methods demonstrated in this video and described in the accompanying guide are detailed in the EPA Methods Manual, Short-Term Methods for Estimating the Chronic Toxicity of Effluents and Receiving Waters to Marine and Estuarine Organisms. Culturing Champia parvula. Although there are 3 macroscopic stages in the life history of Champia, only the mature male and female plants are used in toxicity testing. To keep a constant supply of plant material available, maintain several unialgal stock cultures of males and females simultaneously. New cultures should be started weekly from excised branches so that cultures are available in different stages of development. To guard against microalgal contamination, sterile techniques should be used for culturing the algae. Autoclave all stock solutions and flame all tools before cutting or transferring plants. Keep the cultures on a cycle of 16 hours of light and 8 hours of darkness. The light level should not exceed 500-foot candles. The light may have to be adjusted to that level depending on the reflecting characteristics of the incubators. The temperature should be maintained at 22 to 24 degrees Celsius and the salinity at 28 to 30 parts per thousand. Natural seawater or 50-50 mixture of natural and artificial seawaters are best for cultures. Gently aerate the cultures. Change alternate cultures medium every week so that if a stock solution should become contaminated, the entire batch will not be lost. While replenishing the medium, divide the growing algae in half with sharp forceps, starting new cultures with the cut pieces, or discard half of the biomass to prevent overcrowding. Small branched portions can be transferred directly to fresh media without cutting. Be sure to cool the flamed forceps before touching them to the plants. New cultures can also be started at this time using 1 centimeter plant tips. Add nutrients using a pipette, or, as Narragansett has found, a squeeze bottle is quick and easy to use. Recipes for the culturing medium and nutrient solutions are provided in the supplemental guide. At the end of approximately 3 weeks, there should be enough plant material to conduct the test. Examine the stock cultures to determine their readiness for testing. Cut some plant material using sharp forceps. Place a few branch tips from the female culture in seawater in a petri dish and place the dish under a compound microscope. Male cultures are also inspected under a microscope. Branches from the stock cultures should be placed between the inverted halves of a petri dish. Examine the female branch tips to determine if trichogynes are present. An inverted scope works best with the petri dishes, although standard slides and microscopes can be used. Trichogynes are the short, fine reproductive hairs to which the spermatia attach. They should be seen easily near the apex of the branch tip. Although both sterile hairs and trichogynes occur on the apex, Sterile hairs occur over the entire plant thallus. Sterile hairs are wider and generally much longer than trichogynes and appear hollow except at the tip where they seem to be plugged. Males should be visibly producing spermatia. Sometimes the presence of spermatia sori can be determined by placing some male tissue in a petri dish and holding it against a dark background. Mature sori can easily be identified under a microscope along the edge of the thallus. The sorus areas are generally thicker and lighter in color than the rest of the plant body. At higher magnification, the spermatia themselves can be seen. Conducting the test. Once readiness is established for both males and females, the test can begin. Prepare cuttings from the most healthy-looking plants. Place each plant in a petri dish containing a little seawater. Prepare the female cuttings first to minimize the chances of contaminating them with water-containing spermatia from the male stock cultures. Using fine-point forceps or scalpel, prepare 5 cuttings with the female plants for each treatment replicate, severing the plant about 1 centimeter from the ends of the branch. Try to be consistent in the degree of branching in the cuttings, since cystocarps form at the branch tips. As with the female plants, the males can easily be prepared by placing them in a petri dish with a small amount of water. For male plants, use 1 cutting for each treatment replicate and sever the plant about 2 centimeters from the end of the branch. If there are few branches, larger male plants may be needed. The cuttings can be kept at room temperature for up to an hour. The effluent sample should be kept at 0 to 6 degrees Celsius for no longer than 36 hours before first use in NPDES testing, and never for more than 72 hours. Under a hood, prepare 5 dilutions using a 0.5 dilution factor in 4 100 milliliter replicates. For this test, 3 replicates are the required minimum. However, EPA recommends using 4. At Narragansett, 125 milliliter Erlenmeyer flasks are used as test chambers, but any clean container can be used. The Champia test protocol requires only a small volume of effluent, depending on the concentrations tested. Natural seawater is preferred for the test solutions, although artificial seawater may be used during the exposure part of the test. Maintain the salinity at 30 plus or minus 2 parts per thousand and use colored labels to indicate treatment and replicates. The 2-day exposure period starts when the algae are added to the test chambers. Randomly add 5 female branches and 1 male branch to each chamber. Pick up the branch at the base or cut end to avoid injuring the tips. Cover the chambers with aluminum foil or foam stoppers. In a temperature and light-controlled room, expose the cultures to 16 hours of cool white light and 8 hours of darkness a day for the 2-day exposure and 5 to 7-day recovery periods. Randomize the chambers on the shaker or platform. Maintain the temperature at 25 plus or minus 1 degree Celsius. Also, monitor the salinity in the chambers and maintain it at 30 plus or minus 2 parts per thousand. Check on the chambers twice a day and gently hand swirl or shake continuously at 100 revolutions per minute on a rotary shaker. Spermatia are not motile, so some motion is critical during the exposure period for reproduction to occur. Record the temperature daily from a thermometer placed in a flask of water among the chambers. If desired, the media can be changed after 24 hours. After 48 hours, the algae are removed from the effluent samples and allowed to recover for 5 to 7 days. Label recovery bottles with the effluent concentrations tested and fill with 150 milliliters of natural seawater and nutrients. Smaller volumes should be used with a shaker, but make sure that adequate growth will occur without having to change the medium. With forceps, gently remove all the females from each test chamber and place them into recovery bottles. When all the replicates have been transferred, place the vessels under cool white light and aerate or shake for the 5 to 7 day recovery period. The aeration will enhance the growth rate of plants in the recovery bottles, although adequate growth will occur using a shaker. Aerate using plastic tubes held in place by foam stoppers. Terminating the test. At the end of the recovery period, drain the chambers into a shallow bowl and carefully remove the females with forceps, starting with the control plants and ending with those in the highest concentration. Place the female plants between the inverted halves of a petri dish containing a small amount of seawater and count the cystocarps under a stereo microscope. Cystocarps are distinguished from young branches by the darkly pigmented spores enclosed in the nodule and the apical opening for spore release. If there is doubt about the identification of an immature cystocarp, aerate the plants a little longer in the recovery bottles. Within 24 to 48 hours, the suspected cystocarp will either look more like a mature cystocarp or a young branch, or will have changed very little if at all, indicating an aborted cystocarp. Occasionally, cystocarps will abort, and these should not be included in the counts. Aborted cystocarps are easily identified by their dark pigmentation and often by the formation of a new branch at the apex. Dead plants lose their pigmentation and appear white. The algal sexual reproduction test requires that several criteria be met before the test results are considered acceptable. Control plants should average 10 or more cystocarps, and there should be greater than 80% survival in the control group. Control and lowest concentration exposed algae should be in good physical condition. For example, the branches should not be fragmented. Broken or fragmented branches could indicate that the plants were unhealthy from the beginning of the test. Results from the replicate controlled chambers should be similar. All replicates from the affected concentration chambers should show effect. The methods manual describes the statistical analysis performed on the test results. The red algal sexual reproduction test is currently used to assess the potential toxic effects of complex chemical mixtures on marine and estuarine organisms. Used in conjunction with other whole effluent tests and chemical-specific methods, this test can provide a comprehensive and effective approach to assessing the impact of complex effluents discharged to marine and estuarine environments.